So we were talking about secondary metabolites, so only about their catabolism. We also spoke about that secondary metabolites, they generally have turnover processes. The rate of synthesis also depends on their rate of catabolism or metabolism, which may include the rate at which they are getting transported to the storage sites, or it may also include the rate at which they are getting biotransformed for detoxification and storage subsequently, or directly being used in pathogen attack. So the rate of synthesis is also dependent on the rate of metabolism of the secondary metabolites. So in its interaction with the environment, a plant must always be capable of using its secondary metabolites. So generally we have spoken about this, that at the site of storage may not be the same as the site of synthesis. So it is transported generally through xylem to the parts where it has to be stored, and we know, for example, what are the different ways in which the plant stores it. It may store it in vacuole, it may store it in intercellular spaces, or trichomes or glandular structures, hairs where it is stored, or epidermis or cuticle where it is exposed and where it is needed for the line of defense. Now protective mechanisms have been involved in order for the plant to protect itself from the toxic effect of the secondary metabolite. So what kind of protective mechanism did we talk about? One is biotransformation, like glucose moiety addition, glucosylation. What else? The storage in vacuoles, or keeping the final step where the precursor will get converted to the toxic metabolite only once the cell is damaged. So which means the enzymes of the final step are kept in the vicinity of the precursor moiety. So transport, yeah.
So the transport is of xylem is only unidirectional. So if the secondary metabolite is produced at somewhere at the top, so does the phloem also takes part in the transport?
So it generally depends. Now when we say that at the top is very ambiguous. Now top is everywhere above the ground level. So it is not that it will only happen at one single point of the plant. It will happen aerial. So we can generally bifurcate as aerial or underground parts. So it's a very ambiguous term to even answer that top would mean what. So transport, and there are active as well as passive processes. So active means against the gradient also it can flow. Now transport mechanisms are passive and active processes are involved in transport from the site of synthesis to storage. Now when active processes are involved, which means some transport proteins would be involved. Now upon completion of all possible chemical transformations, they are resecreted in the surrounding medium, which is your xylem or the cell sap. The secondary products are sometimes transferred via xylem to various parts of the plant. Now if you remember, we were talking about induced systemic resistance and SAR, systemic acquired resistance. In both, the signaling molecules were transferred through phloem dependent signal, not the xylem. So I think both are involved in the entire process, in the signal cascade, or the PR proteins flow to different directions, or even for storage of the secondary metabolites transport. Now diurnal cycles and annual rhythms are involved in the secondary metabolite synthesis. Now when we say diurnal cycles, which means that they undergo both temporal and spatial variation. Now this is temporal, which means that the synthesis may be following a 24-hour cycle, or it may be following an annual rhythm, once in a year, twice in a year. So the synthesis takes place. Maybe if it is playing a role in the developmental stage of the plant, then it generally follows the cycles, patterns. Now these variations are caused by the variations in temperature or light conditions in the course of the day. So how do you think that the variations which are following a particular rhythm, the plant is able to maintain these rhythms? By manipulating what? How is the plant managing this? Think logically, don't read. Where is the control?
Synthesizing the proteins only when, like light dependent reactions.
Now everywhere, now these are enzymatic biosynthetic pathways. Enzymes are involved. Enzymes are further controlled by the proteins, by transcription factors. So there are different modes of controls with the plant. The metabolism is controlled. Now by controlling the metabolism, ultimately affecting either the activity of the enzymes or the expression of the enzymes, the plant has the handle to manipulate the rhythms. Now generally in plants you will see that enzyme activity can be impacted by, you know, what are the common factors, by which you think the enzyme activities can get affected. You must have already learnt it. pH, temperature, what else? Light can impact. Now cells in a tissue are exposed to signals from the surrounding tissue. So endogenous chemical gradients can be experimentally induced in cultures for differentiated cells. So generally under in vitro conditions, when we try to do optimizations for maximum production, like for example we add elicitors, which means stress, or we manipulate pH, temperature, RPM. So ultimately it is impacting what? What do we check? If you want to dig down the mechanism, when we add especially the elicitors, yield enhancement mechanism, which means per unit biomass the synthetic capacity of the cell, then we look for the rate controlling steps in the biosynthetic pathway. We see that what is the impact of this particular parameter on any of the rate controlling enzymes, whether on their expression or on their activity levels. Now the possibility of decoupling production and storage capacity, yesterday we were talking about this. Generally it has been observed that these are all intracellular. Generally these are intracellular compounds. The cell produces inside. Now in order to improve the production rates, if you will increase the rate at which it is metabolized or transported outside, will increase the rate of forward reaction, which is the rate of the synthesis. So either under in vitro conditions you have resins to absorb the product, or you decouple the production and the transport to the surrounding medium, or you add permeabilizing agents which can reversibly permeabilize the cell for the product to continuously keep coming out as it is getting synthesized. So these are all among the strategies which we will be discussing, which can improve the productivity of the secondary metabolite in plant cell based bioprocesses. Now the synthetic capacity of dedifferentiated tissue, we know this, often differs substantially from the fully differentiated tissue. We saw that, we were discussing that secondary metabolism in plants is a function of cell differentiation, sometimes morphogenesis, organogenesis. So now it is connected to higher order functions which involves cell differentiation. Now therefore under in vitro conditions you would find that the cells in a callus may not be as productive as if you convert those cells into organogenesis or more differentiated, or if you see that the callus is more compact, there is more strong cell to cell contact, then the secondary metabolite biosynthesis sometimes gets enhanced. Now see, these are the kind of balancing acts between, now if the callus or the cell species is aggregating, which may be good for the yield of the secondary metabolite, but that can be a limitation for, we are improving yield by making it much closer, coming in contact, which would improve organogenesis, biochemical signaling between cell to cell contact would improve, cell differentiation might, the cell differentiation signals might get induced, which is good for secondary metabolite biosynthesis, but where is the limitation? Mass transfer limitations. So the scale up would ultimately get affected, the mixing will get affected, which may cause oxygen gradients. So it is always balancing between the merits and the demerits of any particular strategy. Now production of secondary compounds is usually most efficient in tissues farthest from the meristematic activity. So for higher biomass productivity, you would need that the cells continuously keep dividing. And for secondary metabolite biosynthesis, the word secondary itself says that resting cells would produce better. So, it is a non-growth associated production. So, therefore, decoupling of the production phase and the growth phase is sometimes found to be better in secondary, improving the secondary metabolite biosynthesis. So, for example, like in callus, most of the cells will be parenchymatous in nature. Now, you will see that once the cell are meristematic, they are still expanding, growing, multiplying, and once the cell reaches a matured state, then higher order functions of biochemical activity would appear. So, therefore, it has to be seen that, but for inducing callus, for inducing cultures, you need young explants where there is higher meristematic activity, but production of secondary metabolites, it would be better in matured state of the culture. Now, some of the examples. Expression of hyosamine 6 beta hydroxylase RNA occurs only in developmentally young roots. You will observe that some of the secondary metabolites appear only once the shoot gets up or is induced, or the roots are induced. Till it is in callus or cell suspension phase, the secondary metabolism is not induced. There is no secondary metabolite observed. Formation of small roots in cell aggregates of coleus species induces synthesis of forskolin, a diterpenoid which is of commercial value. Similarly, major components of onion volatile oils are only formed following shoot induction. The onset of cardiac glycoside synthesis in cell cultures of digitalis has been linked to shoot induction. So, these are some of the examples which demonstrate that organogenesis is linked with your secondary, might get linked with secondary metabolism. Nicotiana tabacum callus cultures, root induction is only induced by the addition of nicotine. So, it is two-way. So, nicotine you add and then you see that it has induced what? Root induction or organogenesis in the cultures. Now, plastid metamorphosis also sometimes has been observed to be linked with secondary metabolism, where they have observed in lycopene biosynthesis, where adding a herbicide in tomato cell cultures upon induction of lycopene biosynthesis by the herbicide, what was observed was that the chloroplast started diminishing and they started converting into chromoplast, that red color pigment started appearing. So, in nature what might be happening, that as the lycopene synthesis has to happen for the red thing to come up, the tomato or the fruiting or the maturity of the fruit to happen, the chloroplasts start getting diminished, which means at the proplastid stage, the proplastids rather than getting converted to chloroplast start getting converted to chromoplasts. Now, developmental of chloroplast has, or sometimes it is the other way around, like we are observing in alpha tocopherol as well, that the more green is the callus, the better is the alpha tocopherol yield. So, it is linked to chlorophyll biosynthesis. Even in the biosynthetic pathway, there is a link between chlorophyll biosynthesis and vitamin E biosynthetic pathway. So, similarly, accumulation of cardenolides. So, it is morphogenesis effect. An example, accumulation of cardenolides in digitalis cultures has happened only after the initiation of the green tissues or adventitious embryos. We have observed in Viola odorata also that the cyclotide array or the cyclotide biosynthesis is stable in an organ culture like somatic embryos. Callus, to begin with, when you induce, they show that the cyclotides array is there when it is a fresh callus, but with subsequent years of subculturing, the biosynthetic capacity is lost. So, this is an example which shows us that organogenesis is linked with the, now the same callus, if you convert it into somatic embryos, the cyclotide array is revived. So, these are some of the examples of plant products of commercial interest, where it ranges from pharmaceuticals, then food colors, flavors, fragrances, sweeteners, artificial sweeteners and agriculture chemicals like pesticides or even fertilizers. Now, some of the commercial productions which are happening for the phytochemicals using plant cell based bioprocesses are these. Your anthocyanins, beta cyanins, so these can be pigments, coloring agents, flavoring agents, so there are different kinds. Beta cyanin, carthamin, geranoil, ginseng, health based for good health. Chiconin, it is a coloring agent. Cocoa cells, bilberry cells. So, these are different kinds of products which have been commercialized by Japanese firms, by German firms, by US, by Israelis. So, they are already there in place, are being used for what all uses? If you can see the table, as textiles, as coloring agents, as pigments, as dietary supplements, as nutraceuticals, so as cosmetic agents. These are pharmaceuticals which are getting produced commercially. Anti-cancer, antibiotic, anti-inflammatory, then for the treatment of motion sickness, nausea. So, then pharmaceuticals like berberine, then paclitaxel is well known, it is the Taxol drug which is used for anti-cancer, vincristine, vinblastine, then even your podophyllitoxin. Now, heterologous proteins. Now the interesting part is, it is not only now the plant cells are also replacing CHOs in some cases, where production platforms now CHOs are increasingly used as for production of recombinant proteins, human proteins as vaccines or for animal vaccines or even as antibodies. Biosimilars is one big area. So, where CHO cells are used. Now, there are examples now where some are in human trials, but some plant based these recombinant proteins which have been produced using plant cell based bioprocesses are now approved by FDA and are now marketed. So, some of the examples are your Gaucher's disease, Cerebiose. It is being produced using carrot cell suspensions, got approved by FDA in 2015 and it is now marketed with Protralix. It is an Israeli based company which first made and now it is being sold. Then vaccine against Newcastle disease virus, it was being produced using tobacco cell suspension culture and it is marketed by Dow AgroSciences, and similarly there are many others. So, this is just to show you people that there is so much of scope and the world is gearing up to, and why do you think that it is on the rise? So, one is it is a green technology. So, immune response against the plants, the chances are less. So, it is a sustainable, greener, environmental friendly technology. So, please remember, yield now on would be referring to YP by X, which means the amount of product per unit biomass, and productivity would be volumetric productivity, which is the product titer. Product titer is product concentration per unit time. So, we will be working around in all these strategies, either it will be impacting both the things, the product yield and the productivity, or it will be impacting overall productivity through by improvement in, if it is not impacting product yield, but still the productivity is high, can be because of time. So, from where is that coming? Probably, generally these are intracellular products. So, these are generally intracellular products. So, when we say product titer, what does it mean? This will include what two parameters? So, from where is this amount and volume coming? When we say productivity in an intracellular based product, it will be suppose grams per liter per time. Now, this gram per liter per time is a club of what two things? How do you measure gram per liter per time? How would you measure? Think that ways.
After all the extraction, the amount of product we got and total volume before the extraction, from the total volume.
So, how do you get the total amount of product?
From different concentration.
What concentration? Think a little math is involved. how would you calculate? Start from the scratch. I said product productivity would be, assuming it is an intracellular product. So she said it will be extracting from the biomass. So how will you calculate? How will you get 2 grams per liter per unit time?
Total amount of biomass which we had initially is will come in denominator, and after all the extraction steps,
So for extraction, every time you will have the entire biomass for extraction? How will you do it at a experimental level? Think that ways. You are running a shake flask or you are running a reactor. What would you do? You will every time harvest the entire biomass and then you will dry it, then you will extract using the entire biomass?
no, that's this. If in 1 ml I have 1 mg,
Okay.
it is 1 mg per ml. So what would be there in 1 liter? So it will be
Right. So here 1 ml would be there. Ml is what then?
ml is cell biomass, the total mass.
Will it be volume, cell biomass?
Okay, it will be weight by weight.
Weight by weight. Now this weight by weight is called what?
Yield.
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 So one is yield. The other would be
Rate of production.
rate of what?
The reaction time.
The mu.
So this mu is specifies what?
The kinetics of the
Of
reaction, production of biomass group.
so now it will take care of your, now this was, if you think mathematically, it is gram per gram biomass multiplied by the concentration of the biomass. So this will give you the product titer, isn't it? And then the time in which that product concentration or the biomass is achieved. So that is what is overall volumetric productivity. When you have mu, it becomes what is it called? When you have mu, what will it be called?
Biomass productivity.
Along with the yield, what will it be called? Think about it. It is simple maths. I want you to represent your product after evaluation as the volumetric productivity or the product concentration. How would you determine? Just think on those lines, then you will be able to figure out that what should be your 2 important responses when you are optimizing a process. What will be your 2 objective functions? Okay, so we'll continue tomorrow.